Welcome to Biology, the scientific study of life. Toxins in the environment. Humans release an immense variety of toxic chemicals, including thousands of synthetics previously unknown in nature, with little regard for the ecological consequences. Organisms acquire toxic substances from the environment along with nutrients and water. Some of the poisons are metabolized and excreted, but others accumulate in specific tissues, especially fat. One of the reasons such toxins are particularly harmful is that they become more concentrated in successive trophic levels of a food web, a process called biological magnification. Magnification occurs because the biomes at any given trophic level is produced from a much larger biomes ingested from the level below. Those top-level carnivores tend to be the organisms most severely affected by toxic compounds in the environment. One class of um, industrially synthesized compounds that have demonstrated biological magnification are the chlorinated hydrocarbons, which include many pesticides such as DDT and the industrial chemicals called PCBs. Polychlorinated biphenyls. Current research implicates many of these compounds in endocrine system disruption in a large number of animal species, including humans. Biological magnification of PCBs has been found in the food web of Great Lakes, where the concentration of PCBs is inherent gold eggs at the top of the food web is nearly 5,000 times greater than in phytoplankton at the base of the food web, as shown in this figure. An infamous case of biological magnification that harmed top-level carnivores involved DDT, a chemical used to control insects such as mosquitoes and agricultural pests. In the decades after World War II, the pesticides industry promoted the benefits of DDT before anybody really understood the ecological consequences. By the 1950s, scientists were learning that DDT persists in the environment and is transported by water to areas far from where it is applied. But by then, the poison had already become a global problem. On the first signs that DDT was a serious environmental problem was a decline in the population of pelicans, ospreys, and eagles, birds that feed at the top of food webs. The accumulation of DDT and DDA, a product of its partial breakdown in the tissues of these birds, interfered with the deposition of calcium in their eggshells, a trend that may have already begun because of other environmental contaminants. When these bears tried to incubate their eggs, the weight of their parents broke the shells of affected eggs, resulting in catastrophic declines in their reproduction rates. Rachel Carrinson's silent spring helped bring the problem to public attention in the 1960s. And DDT was banned in the United States in 1971. A dramatic recovery in populations of the affected bear species following the pesticide is still used in many other parts of the world. However, many toxins cannot be degraded by microorganisms and consequently persist in the environment of years or even decades. In other cases, chemicals released into the environment may be relatively harmless but are converted to more toxic products by reaction with other substances or by the metabolism of microorganisms. For example, mercury a byproduct of plastic production and coal-fired power generation has been routinely expelled into rivers and the sea in an insoluble forms. Bacteria in the bottom mud convert the waste to methyl mercury, an extremely toxic soluble compound that accumulates in the tissues of organisms, including humans who consume fish from the contaminated waters. Atmospheric carbon dioxide. Many human activities release a variety of gaseous waste products. People once thought that the vastness of the atmosphere could absorb these materials without significant consequences. But we know now that such additions can cause fundamental changes in the composition of atmosphere and its interactions with the rest of the biosphere. One pressing problem is the rising level of atmospheric carbon dioxide. Rising atmospheric CO2. Since the Industrial Revolution, the concentration of CO2 in the atmosphere has been increasing as a result of composition of fossil fuels and the burning of enormous quantities of wood removed by deforestation. Scientists estimate that the average CO2 concentration in the atmosphere before 1850 was about 274 ppm. In 1958, a monitoring station began taking very accurate measurements on Hawaii's Moyana Low Peak, where the air is free from the variable short-term effects that occur near large urban areas. At that time, the CO2 concentration was 316 ppm, as shown in this figure. Today, the concentration of CO2 in the atmosphere exceeds 370 ppm, an increase of about 17% if CO2 emissions continue to increase at the present rate. By the year 2075, the atmospheric concentration of this gas will be double what it was at the start of the Industrial Revolution. Increased productivity by vegetation is one predictable consequence of increasing CO2 levels. In fact, when CO2 concentrations are raised in experimental chambers such as greenhouses, most plants respond with increased growth. However, because C3 plants are more limited than C4 plants by CO2 availability, one effect of increasing global CO2 concentration may be the speed of C3 species into terrestrial habitats previously favoring C4 plants. This may have important agricultural implications. For example, corn, maize, a C4 plant, and the most important grain crop in the United States may be replaced by wheat and soybeans, C3 crops that outproduce corn in a CO2 enriched environment. To predict the gradual and complex effects of rising CO2 levels in species composition in non-agricultural communities, scientists are turning to long-term field experiments.